Intermembrane space.
Electron
ATP.
Okay, so I made a mess here of my board. All right. So, inner membrane, outer membrane, intermembrane space. Inter means between, by the way. Did you know inter means between? Inner membrane, outer membrane, intermembrane space. Matrix. What are the folds called? Cristae. What do the folds do? They increase the surface area. So you can fit more of these proteins on the inner membrane. These proteins are called the electron transport chain proteins. And they line the inner membrane here. That's why it's highly folded. So you can fit more of these proteins, and that's where the electrons are going to be transported along here so we can make ATP. The whole purpose of the mitochondrion is to make ATP. Okay?
Okay.
So, how does this occur? How does the breakdown of glucose occur? Well, it starts with a process called glycolysis. Can you say glycolysis?
Glycolysis.
All at the same time?
Glycolysis.
So, glycolysis, we take glucose. There's glucose at the start. I have a glucose molecule up here. Bam. And glucose, of course, do you know the formula for glucose?
C6H12O6.
C6H12O6. That's glucose. It's got 6 carbon atoms. I only see 1 carbon atom in my molecule here. Where are the others?
Where
the lines intersect.
Where the lines intersect. Well said. There's a carbon there, there's a carbon there, there's a carbon there, there's a carbon there, there's a carbon there. So, there's 6 C6, 12 H's, and 6 O's. That's the glucose molecule. Now, I'm going to represent it during the next 4 or 5 days like this. These are carbon atoms. I'm going to put them together in kind of a hexagon pattern. So this is that. Can y'all handle that?
Yes, sir.
Now I'm only showing the carbons, but there are actually 12 hydrogens and 6 oxygens also in this molecule. But I don't have room for them up here. I mean, I could put 12 hydrogens if that's what you want, if you're going to get so upset about it. I could put 12 hydrogens. But that will make it a little hard to move around, don't you think?
Mm-hmm.
So you're just going to have to imagine that those are there. And I could also put 6 oxygens on there. I've got some oxygens. But as you can see, the molecule is now getting out of hand. I'm not going to be able to move it around. So can you just imagine that they're there? You have a decent imagination?
No.
So, that's what we're going to go with. That this glucose molecule has a lot of stuff in it. And not only does it have hydrogens and oxygens, it also has a lot of electrons in it. And I'll be using electrons. These electrons are involved in the bonds. Every little line you see there is a bond between atoms. There's a carbon there, there's a carbon there, there's a line between them. That line between them is a bond, which is held together by shared electrons. You remember they share their electrons? So we're also going to be using these electrons, and you'll see them falling out of this molecule as we break it down. Chandler, are you listening? You seem like you're staring at my shoes. These shoes are pretty good. I can move pretty quickly in these shoes. Well, you really don't want to see it. It's fantastic. There's not enough room in here to really show you the full acceleration. So, let's break down glucose. You want to?
Heck yeah.
If we're going to break down glucose, you need to know that whenever you break down a large molecule, I like to say that when we take a big molecule and we break it apart, like here's glucose, what if we broke it in half like that?
Whoa.
Whenever you break apart a big molecule, stuff flies off of it. I always use the analogy of Godzilla. I always think about Godzilla. He would pick up, you know who Godzilla is? It's a big dinosaur from Japan, and he grabs a bus or something, and he just tears it in half. And it'll explode whenever that happens. And people come falling out, and glass goes everywhere, and the doors fall off. Whenever you take a big molecule and break it apart, stuff's going to fly off of it, right? Keep that analogy in mind. Because when we take glucose and we tear it apart, stuff comes out of it. And that stuff is electrons and protons. Did you know that the hydrogen ion is a proton? Have I shared that with you? Because a hydrogen atom is just a proton and an electron. And if you tear the electron off, you end up with just the proton, which is the hydrogen ion. So whenever you tear apart a big molecule like this, you're breaking bonds, and the electrons that were there sharing the bonds can come flying off, and the hydrogen ions can come flying off too. And what we're going to do, this is a very simplified thing. Glucose cannot just be directly torn apart like that. But if you, in a simplified fashion, if you think about it this way, if this is torn apart, whoops, and these protons and electrons come flying off, they can get picked up by an electron carrier like NAD. NAD is an electron carrier. It picks up electrons, it picks up protons. And we form NADH at the end. This actually happens twice during glycolysis. When we tear glucose in half, we end up forming two NADHs. So another NAD comes and picks up protons and electrons. And also what we're going to form is a couple of ATP molecules. So when we tear this glucose in half, energy comes out of it, and the energy is used by an enzyme to put ATP together by substrate level phosphorylation, which is what you read about. Substrate level phosphorylation basically works like this picture here. Here's an enzyme. Come on. Here we go. Here's an enzyme. Here's ADP, which is this red molecule. Here's some substrate that had like a protein or something that has a phosphate connected. We'll pretend it's this thing right here. It's got a phosphate on it. And what happens is there's an enzyme. I'll draw the enzyme. Let's say it's right here. And it's kind of shaped like this. Oh, that's great. I did a great job. And so the substrate with the phosphate attaches here. And the ADP attaches there. I didn't do such a great job. Close enough. And then it's going to transfer the phosphate over, and they're going to come off. That's substrate level phosphorylation. Did you see it? Why do we call it substrate level phosphorylation? Because there's an enzyme and a substrate. And the substrate is supplying the phosphorus to ADP to make ATP. So it's substrate level phosphorylation. And that's how ATP is made in glycolysis. Glycolysis is the first reaction of tearing down glucose. And that was just the short of it. We create 2 ATPs by substrate level phosphorylation, and we create 2 NADHs by adding H and electrons to NAD. Now, it actually happens in 9 steps that they go over in your book, but they aren't important for you to know. You won't be tested on the steps of glycolysis either on my quiz or on the actual test. You don't have to know all these intermediate steps, but it does talk about them in your reading, but you don't have to know them all. Now, if it did show you a picture, like if it showed you this picture, it might ask you a question about the picture. But you would have to memorize all these names in the middle. You could look at the question and look at the picture and answer questions about it, though. That might happen. So look at this. We start with glucose. The glucose is changed into glucose 6-phosphate. Do you notice that we go from a ring formation to a straight line?
Yep.
Yeah.
We unhooked, we broke one of these bonds, so it's no longer a circular formation. It like straightens out. That's what happens in the first step. We added a phosphate to it. So what if I asked you, where did that phosphate come from? And you're allowed to look at the diagram. Glucose got changed to glucose 6-phosphate. You can see this phosphate added to the last carbon. Where did the phosphate come from? Can you look at the diagram and figure it out?
ATP.
Well, there's hexokinase. And ATP loses a phosphate to hexokinase, and then hexo whatever puts the phosphate on the glucose.
So where did the phosphate come from?
ATP.
It came from ATP. I heard you, Lottie.
Yeah,
I mean,
Lottie.
I was wanting Carter to finish formulating her answer because I thought she was onto it, and she was. An ATP. Notice this ATP changes to ADP, and this phosphate is given to glucose. Now you have glucose 6-phosphate. So basically, what we did first is we did this. Watch this. Straighten that thing out. Isn't that fun? Now, I thought, Mr. Willis, I thought you said that we make ATP in glycolysis. That wasn't making an ATP. That was using an ATP. We do. We make ATP later. This step, the first step of glycolysis, actually there's 2 phosphates added. Do you see the phosphate is added there in that step? And another phosphate gets added here in step 3. Now there's 2 phosphates connected to the molecule. It's no longer called glucose at this point. It's called fructose 1,6-biphosphate. A phosphate's been added here and a phosphate's been added here. We actually are going to add 2 phosphates to the structure in the first part of glycolysis. And that uses up 2 ATPs. This is called an energy requiring step. We've actually lost energy here. We're doing the opposite of what we need to do. We're not making ATP. It starts out by giving up ATP. Sometimes you'll see questions that say that this is like activation energy. Y'all remember activation energy in a chemical reaction? You have to put in energy to get something to react, and then it goes downhill and gives off a lot of energy. Y'all remember that? Think about lighting a piece of paper on fire. You actually have to put in energy first, and then the paper gives off a whole lot of energy, right? When it burns. So this is like activation energy. We put in 2 ATPs first to change the molecule to something. And after it forms this, there's an enzyme that will break it in half. So this molecule gets broken in half. Bam, bam. We form these 2 things. They're called dihydroxyacetone phosphate molecules. Well, one's called dihydroxyacetone phosphate, and one's called glyceraldehyde 3-phosphate. But anyway, what we're basically doing is this. An enzyme comes in, and it breaks this in half like that. Now we're at this step right here. And what happens next? Well, here, an NAD comes in and picks up some hydrogen, a hydrogen and an electron to form NADH, hydrogens and electrons. So here's where now we got these molecules, and here's where the NAD comes in and picks up stuff. And we form that NADH. And this happens twice. See, it says 2x, 2 times? So it happens twice. So 2 NADs come in and pick up electrons and hydrogens from the structures and make our 2 NADHs. There's where we form our NADHs. Right? Come on. Where's my... Hello. There it is. So NAD comes in and forms NADH. Now, the NAD actually brought in a phosphate as well. So, I didn't show that. When the NAD originally came in, I guess I should show that. When the NAD originally came in, it dragged a phosphate with it. So it adds a phosphate, and then it takes the electrons and the hydrogens. And that happens to both of these molecules. Again, I won't make you remember all this, but I'm just trying to show you this diagram and explain it. If I had any questions about this, I would show you the diagram. Okay? Now, here's the fun part. Here's where we actually make ATP. You see how this molecule has 2 phosphates on it?
Mm-hmm.
It's called 1,3-biphosphoglycerate. It's called biphospho because it's got 2 phosphates, one there and one there. And this thing, now we're going to lose one of the phosphates and make ADP into ATP. So here we come. There are 2 phosphates on this one. There's 2 phosphates on this one. So ATPs are going to come get one of the phosphates in this next step. We make 2 ATPs there. And notice now we've lost this new molecule, 3-phosphoglycerate, has lost one of the phosphates. We made some ATP. We make 2 ATPs there. And then down the road, we lose a water molecule. The last phosphate gets removed, and we make 2 more ATPs. So 2 more ADPs come in. They take the last phosphate. So we started by giving up, by breaking down 2 ATPs. We finished by making 4 ATPs. What's our net gain? 2 ATPs. We use up 2 ATPs, but we make 4 ATPs. So you have a net gain of 2 ATPs. So if you want the short version of it, I like doing the short version, especially in my regular biology classes. Here's glucose. Bam! It gets torn apart. Stuff flies off. We make 2 ATPs from the energy that comes out. And we make 2 NADH2s from the protons and electrons that come out. That's the short version.
Is it always torn apart in 3 carbons?
Yes. It's always torn apart in 3 carbons. And what do we have at the end? We have this molecule, and you do have to know this name. we start with glucose, we end up with 2 pyruvates. 2 pyruvates. Here's another thing you need to know. Whenever you see 8 at the end of an organic molecule, you can substitute ic acid. 8, the prefix 8, is the same as the prefix ic acid. So, pyruvate is the same as pyruvic acid. What if I told you something was called citric acid? Have you ever heard of that?
I have.
What else might you call it? Citrate. And so this is phosphoenol pyruvate, which is one of my favorite molecules. I don't know why, I just like to say it. Phosphoenol pyruvate, what might you call it? Phosphoenol pyruvic acid. So there were 10 steps in that glycolysis to get from glucose to pyruvic acid or pyruvate. Would you like to see video footage?
Yes, sir.
You don't have to memorize all the 10 steps, but when I was in college, they made us know all of them. And I had a buddy that was a friend of mine that had this on a poster in his dorm room. Hey, good grief.
Glycolysis, which begins the breakdown of glucose, is a series of 10 enzyme-catalyzed chemical reactions that can be divided into 2 main phases. In the energy investment phase, some ATP energy is used to start the process of glucose oxidation. By the end of this phase, a 6-carbon molecule, glucose, has been split into 2 3-carbon molecules of glyceraldehyde phosphate. The 3-carbon glyceraldehyde phosphate molecules now enter the energy payoff phase. Chemical bonds are broken, and NAD+ picks up electrons and hydrogen ions, forming NADH. The energy released is used to attach phosphate groups. The phosphates are transferred to ADP, finally making some ATP. This way of making ATP is called substrate-level phosphorylation. A couple more reactions rearrange the atoms in the 3-carbon molecules. More ATP is generated in the final reaction that yields pyruvate. For each glucose molecule broken down during glycolysis, a net of 2 ATPs are formed, along with 2 NADH molecules.
How was that? Now, this is the only way your cells have of making ATP that doesn't require oxygen. All other ways of making ATP happen in the mitochondrion of your cells. Every one of your cells has mitochondrions living in it. Well, I say living because they were once free living organisms. Do you guys remember that?
Yes, sir.
That were taken into cells by endosymbiosis. And they're really just kind of crawling around inside your cells. They have their own DNA. They have their own ribosomes. It's just like a bacteria that's invaded your cells. So I like to say that they're living. But anyway, this mitochondrions, every one of your cells has, could be 100, could be 1,000 of these in a cell. Maybe 30 or 40 cells that don't do much, don't need many mitochondrions. But to make more ATP, these pyruvates have to go into the mitochondrion, actually into the matrix of the mitochondrion, where they'll be further broken down. But you have to breathe oxygen for your mitochondrions to work. The mitochondrions do not work without oxygen. So if you don't have oxygen present, the only way to get ATP is through glycolysis. Now, whenever you're doing a quick exercise, like a sprint or something like that, You don't have time to make the ATP with your mitochondria. The mitochondrions work relatively slowly. The fast way to make energy is to break apart the glucose out here, outside of the mitochondrion, in the cytoplasm of the cell. And you can make, and that can happen, this glycolysis reaction can happen very fast. It goes through those 10 steps, and it's done, it's broken down, and you make some ATP, and this is called anaerobic respiration. Y'all know what that word anaerobic means?
Without oxygen.
Without oxygen. An means not aerobic. That's without oxygen. And your quick activities, your sprints, when you're really just trying to lift as much weight as possible, it's happening really fast, and it takes this, it makes the glucose into pyruvate, and then the pyruvate is converted very quickly into what's called lactic acid, and that's what makes your muscles burn. Now, in time, the lactic acid will move into the mitochondrion and be broken down, but that requires oxygen. That's why after a sprint, you're breathing real heavy. You're trying to get oxygen to your mitochondria to break down these products, the products of glycolysis, and that takes a little time. Isn't that interesting? We're seeing how muscles work. Why are we showing bacteria? Oh, because bacteria, prokaryotes, don't have mitochondria. The only way that bacteria can make energy is through this glycolysis. As a matter of fact, every organism on Earth does glycolysis. All bacteria, all archaea. It is a universal process for making energy. So we think, since every living thing does it, prokaryotes, eukaryotes, everything, we think it was one of the first processes that evolved in living things. If everything does it, it must be really old. So, that's glycolysis, baby. So, what we're going to do next time on Monday is we're going to learn about what happens next. And let me just show you the steps. You can remember the steps of the breakdown of glucose by this acronym. Good people can excel. Good people can excel. Glycolysis, the preparatory reaction, the citric acid cycle, and the electron transport chain. Four steps on the way to tearing glucose apart and making ATPs. Do you know how many ATPs we'll make for each glucose molecule we break down?
18.
How many ATPs we'll put together from the energy of the breakdown of one glucose molecule?
9.
36. Okay, this was today, Friday, Monday, Tuesday, Wednesday. This is what's in store for next week.
Alrighty.
Exciting?
Very.
Willis.